This program is brought to you by Emory University.
All right, so on to today's lecture. Phytochemistry and contemporary drug discovery, and what all that means. So today we're going to basically discuss the regulation of pharmaceuticals, botanical drugs, and dietary supplements, how they're similar, how they differ. We'd like to describe what bioactivity fractionation and structure elucidation are, which are basically what I do for a living, and what's done in the lab right next door that everybody kind of walks by and doesn't know is a lab. And then I'm going to run you through a whole lot of chemistry tools. Basically, you're going to get a semester of analytical instrumentation slammed into you today. I promise there won't be many formulas and there won't be a whole lot of math, so it'll be the very high-level overview, but I would like y'all to be able to understand what are these big tools and how are they used. And I think that gets into why it costs so much to develop drugs, to understand how many different pieces of technology are wrapped up in this these days. All right, so who am I? That's me. I'm a natural products chemist and a pharmacognosist. I stole this slide right out of my 9th grade talk last week that I gave to middle schoolers. So my PhD is in biology with essentially a focus on the plant sciences. I worked at the New York Botanical Gardens and the City University of New York. So I came out of a very big plant program, lots of taxonomists, lots of systematics kind of thing. After that, I worked for a couple of years up in North Carolina and Asheville as an extraction chemist and analytical chemist. And part of my job was making lots of plant extracts. The other part of my job was analyzing dietary supplements and botanical ingredients for safety. So we'll talk, that's where the regulations are about to come up. Currently, I'm a postdoc over in Dr. Quave's lab, which basically means I do everything that's asked of me. If it's chemical, it probably comes my way. And the one sentence takeaway from this for the 9th grade level understanding of what I do is I solve chemical problems that nature tosses out. I tossed two big words at you. One of them was pharmacognosy. It is the study of chemical, mainly, is what I'm interested in, the chemical properties of potential drugs of natural origin. You'll notice it doesn't limit you to plants. People also work on marine invertebrates and tunicates, whole sorts of things. Fungi call into this list. The other part is, for the plant section, plant preparations are medicinal or herbal when they're used to promote health beyond basic nutrition. So everybody in here eats their vegetables and grows up to be a big, strong boy and girl. That is not herbal medicine. That is not a medicinal plant. Those are food, essentially. Even though dietary supplements are regulated as food, the differentiation for me is when you start using your food to treat things. Health, either make you a healthier person or to treat an ailment. That implies that you have some type of knowledge, be it traditional or Wikipedia-based. You have some knowledge to apply these plants to an ailment. Really need a clicker in here. All right, so skipping ahead, or skipping around a little bit here. Pharmaceutical development cost currently, it's $802 million. That's per drug. That's the equivalent of 40 F-16 jets in 2003 dollars. That's an F-16 in the background, just to remind you, Top Gun-esque. It is also more than the gross domestic product of all of these countries, according to the World Health Organization. So it is more money than that entire country takes in a year. And those aren't necessarily small countries. By the way, the U.S. is way, way up there in trillions of dollars. So. It's a lot of money to develop a drug, and it takes a lot of time. Currently, the pipeline is somewhere between 8 and 12 years, and that is from initial lead, as in I have a compound that I think can make a good drug, to when I can go to the store and get it by prescription. That is 8 to 12 years. There are rapid ways that it can be done now. Sometimes if you work on cancer or HIV especially, where the mortality rate is extraordinarily high, that can be shoved down to about 3 to 5 years because the patients don't live 8 to 12 years. So there's a much quicker cycle for the approval. But by and large, you're looking at a decade. Part of the reason it costs so much is because the winnowing down of potential drugs to actual prescriptions is very, very steep. This is from a paper in 2007. So in 2007, 30,000 drug candidates went into the drug development pipeline. The numbers on the right give you an idea of how many make it to kind of each stage. Of the 300,000, sorry, not 30,000, about 21,000 made it past the initial screening stage, and it was like, okay, these can be drugs. Of those, only 380 made it to phase 1 trials. Phase 1 FDA trials are the ones that are the first time it's done in people, and we basically give it to you for safety. You are a healthy individual, we give you our potential drug, and we see if you vomit, we see if you get diarrhea, get cramps, go blind, die. It's a safety study. By then, you probably are not going to die, but that's where that long laundry list of possible side effects with every drug comes from. It's the phase 1 study. It's what healthy people get when they take your drug. You can see it just drops down further and further. This is an estimate of, in 2007, what was left. They expect 23 new drugs. That's drugs for everything. Everything from hurt muscles to cancer to HIV to restless leg syndrome. So out of 300,000 hits, 23 drugs will be approved. That is a huge investment for a pharmaceutical company and a whole lot of failing. So that's why it costs so much. They have to recoup a lot of failure for every drug that gets approved. The old joke among medicinal chemists is, Tylenol only cost you a penny a pill, right? But that first pill of Tylenol that they cost, well, it cost $800 million. So the first one's $800 million, the second one's a penny. So that's kind of the way a pharmaceutical company has to think about this. We're going to look at a lot of text. You don't need to know all of this text. I tried to highlight what's important, but this is the Food, Drug, and Cosmetics Act. This is where drugs are defined and regulated in the U.S. code. That's the U.S. legal code. We'll also look at dietary supplements. So the important part here is articles intended for use in the diagnosis, cure, mitigation, treatment, and prevention of disease in man and or other animals. That's what a drug is, according to the government. There's a couple other things in there that are interesting little tidbits. If it is recognized in the United States Pharmacopoeia, the Homeopathic Pharmacopoeia, or the National Formulary, these are books that predate the FDA. The FDA was founded in, I think, the 30s or 40s. I have it on one of the slides coming up. These were essentially herbal texts and medicinal books, like big encyclopedias with formulas that predated the FDA. If they were in there already, then they got approved as a drug now. They just kind of got grandfathered in. Most countries have a pharmacopoeia, and they are now updated, so every new drug kind of gets added in there, and that's where the specifications for purity and use are actually put. Let's see. That's the important part on that slide. The key difference in making something a drug versus other things, they are generally recognized as safe and effective. That and effective is the key portion of that phrase, safe and effective. And the benefits must outweigh their potential risks. So the safe and effective part, I'm a pharmaceutical company, I do all sorts of tests, I produce tons and tons of data, and I say it is safe, and here's the science that I have done to say that it does something. And that goes to the FDA. And the FDA says, yep, we think your science works, or your science supports the premise that this drug does what you think it does. And then they also have to report all the side effects and all the adverse reactions to the drug to the FDA with that. And the FDA looks at that and says, well, your drug saves lives and only 1% of people end up vomiting for 6 to 12 hours. We think saving lives versus vomiting for half a day is a good use of this drug. So the benefits outweigh it. If it saves lives but 50% of the people die, then they might not approve it. That is a judgment call that's made at the FDA level, not by the company that's actually doing the research or putting the drug up. The three big acronyms in the alphabet soup world you'll always hear and you'll always see with any drug company or any drug discovery program is IND. This is an investigational new drug. And this gets into a little bit on how drugs are developed. The investigator, i.e. the scientist or the company that is developing the new drug, they will submit to the FDA, I want to take this compound and test it in people to alleviate motion sickness. And the FDA will say, okay, that seems like a good use. There's some data to support this. You can go ahead and do that study. And it's just a study. One compound, one use. New drug applications. So once I've done all the phase 1 and phase 2 and phase 3 and all the trials and all the science, and it's literally 30, 40, 50, 100 boxes of files, they still do these in paper, get shipped to the FDA. And that is a new drug application. You say, I would like to sell this drug for this ailment X to this population Y. And here is all of my data that says why it's safe and why it's effective. And then the FDA goes through all that, and they will either approve your application or not approve your application. And the last thing is an active pharmaceutical ingredient. This is the single compound or mixture of pure compounds that are actually responsible for the activity. There are other things in every pill. The actual capsule is usually made out of gelatin, the same stuff as your Jell-O. There's excipients in there that make it easy to flow and stay in a powder. There's fillers, there's additives, there's whole sorts of things mixed in there. But the one that actually matters as far as the benefit is called the API. All right, U.S. Dietary Supplement Regulation. So this is completely different than drugs. They are regulated as a food. The Food, Drug, and Cosmetic Act was in 1938. That was when the FDA basically was founded. It was revised in the 70s, in 1976, but it's not until DSHA comes around, the Dietary Supplement Health and Education Act in 1994, that the term dietary supplement, as you know it today, is actually defined. So that's a fairly new thing. It's an amendment to the FD&C, so it's just an add-on to the law, but everybody just refers to it as DSHA. All right, so the term, as it's defined in the code, this is U.S. code, whatever, 21 blah, blah, blah. You can look it up, but here's the text. Dietary supplement is an herb or other botanical, concentrate, metabolite, constituent, extract, or any combination of those ingredients. This is key, intended for ingestion. It can't be eye drops. It can't be suppository. It can't be an injectable. It can't be a patch. It has to be something that is consumed for it to be a dietary supplement. And it has to be labeled as a dietary supplement. So there's a labeling issue here too. If I say it's a dietary supplement, automatically it starts out as a dietary supplement until it's ruled otherwise. You, the manufacturer, establish specifications for everything that's in there. I tell you what's in it, and I tell you what levels of contamination and adulteration I will allow in my product. Me, the manufacturer, me, the guy who's here to make money on the product, is the one who establishes what can be in there and what can't. And I must establish these specifications with at least one appropriate test to determine 100% identity of the ingredient. This is my favorite part. Using an appropriate scientifically valid method. Such examples are these. Me, the manufacturer, can determine what is a scientifically valid test to prove that my dietary supplement has what should be in there, and I only have to do it with one analytical test that is scientifically valid by my definition. So at this point, you should have a couple flags going up in your head. Who's making the dietary supplement specification? This is a hands up kind of question and answer session. Anybody? Somebody muttered something. Or Micah?
People who are going to sell it?
People who are going to sell it. So the people responsible for establishing the dietary supplement specification when it's actually sold to the public initially are the manufacturers or the people selling it, not any regulatory body. That's not to say there aren't regulatory bodies that can come in and then disagree with you, but initially it's the manufacturer, the guy who's making a profit out of it. This is also the same in pharmaceuticals, right? Who's initially saying all this data is safe, all of our data says our drug is safe and effective? It's the pharmaceutical manufacturer. But that is all reviewed by the FDA before it's allowed to go into the public. Dietary supplements, I just submit it all to you and I can start selling as soon as the letter is received. Does a dietary supplement have anything to do with effectiveness? Nope. It just has to do with safety. I can put sawdust in a pill and sell it to you as goji berry. And as long as it doesn't kill anybody, I can sell that until somebody stops me.
Just for clarification, are we making a distinction between pharmaceutical, drug, and dietary supplement?
At this point, yes. We'll bridge them in one slide. So dietary supplements are regulated as food. Pharmaceuticals are regulated as pharmaceutical drugs by prescription. One can become the other, but their regulation is very specific, and they end up in one bin or the other when they get to be sold. So who establishes the safety of the dietary supplement initially? Again, the manufacturer says it's safe. And how does he do it?
One test.
One scientifically valid test. Is testing expensive? Hint.
Yes.
It's expensive. So me, the manufacturer, by and large, there are good dietary supplement manufacturers who have full analytical chemistry labs and do the right thing. But there are also ones that just pick the cheapest thing out of the catalog and say, oh, that works. It may or may not be the best test. So these are all things to keep in mind when you go to buy this sort of stuff. Just to give you a quick comparison, in Germany, everything's regulated very differently, and in Europe. So the German Commission E, it's in 1978, it was basically the same thing as our FDA in between 84 and 1994. They went through and just looked at all these herbal products, and they created monographs for each of them, basically licensing 380 different herbal products as prescription drugs. So doctors can prescribe them. The formulation is standard. It's in a book. Everything has to be approved when you sell it to the German Commission E standards, which is very different than the way we regulate things, right? The manufacturer makes the regulations here. All right, we're going to go this way. So here's our quick example. This is why your generic Tylenol will be fine, but your diet pill may kill you, part of the lecture. Generic Tylenol, right? Tylenol, Walmart brand. What's the difference? Branding. One's $3, one's $9. They're the same pharmaceutical ingredient, the same API. They're both regulated by the FDA as the same drug. They're both manufactured according to GMP, GLP. The difference is just who's making the money on it. The patent is expired on Tylenol, so it can be made as a generic, and other companies can get in on the action, so to speak. Dietary supplement adulteration. So this is Super Arthro Gold. It was pulled from the market in 2014 by the FDA. It was, by the company, sold as an all-natural source formula that is known to help with arthritis, joint, and muscle-related aches and pains, essentially a Tylenol alternative. It worked great. Problem, it contained 3 prescription drugs. One's a muscle relaxer, I guess 2 prescription drugs and 1 over-the-counter, and 2 NSAIDs, essentially Tylenol-type drugs. When they're mixed together, you can die. FDA ordered it recalled. So it was very effective, just not safe. That's why it was pulled from the dietary supplements market. This is my favorite one, probably of all time currently. This is Superpower, Ride Through the Night is the tagline written up there. It was sold in truck stops and gas stations as a male enhancer. It is an all-natural herbal extract. If you look here, this is the back of the package that tells you all sorts of stuff. This is weird botanical Latinese. Semen just means seed. In this case, not something else. But it's got cinnamon, cassia, that's cassis cinnamon. There's some ephedra in there, ginseng, gelatin, all this stuff. Turns out it worked great. It sold like hotcakes. It was recalled voluntarily by the manufacturer after a letter from the FDA because, well, it worked well because it contains sildenafil. Anybody know what that one is? Oh, come on. It is Viagra. It was adulterated with Viagra. It worked probably excellently, but if you're taking it as an herbal supplement, you're on, I don't know, nitrates or any of the other lists of things that are not supposed to be taken with Viagra, you're going to have some problems. So, pharmaceuticals, dietary supplements. This is kind of the bridge between the two. A botanical drug. The FDA has established new guidelines, new about 10 years old now, for botanical drug products because they are often complex mixtures that lack a distinct active ingredient and have a history of prior human use. So you can take a botanical and make it into a drug now, or you can take a dietary supplement and make it into a drug. There is a pathway to do it. It's been done 3 times, but it does exist. What does this look like as far as the chemistry is concerned? This top chromatogram, this is actually Tylenol. This is acetaminophen right here, Tylenol, and this is one of its, it's a cysteine analog, so ignore this one, but that's what's in Tylenol. This is one of the ones we work on in our lab. Anybody see a difference? One thing in the top one, there's 42 things in the bottom one. Little different regulatory pathway, a little bigger pain in the rear to work on, and a whole lot harder to identify a single compound for activity. And this is where we get into the chemistry part. So because we have so many things in a botanical dietary supplement or a botanical drug, we have to start separating them out and detecting and understanding what they are. So separation, unmixing. The picture on my left, your left as well. It's real easy to separate the rocks from the water, right? We have filter paper, it does that. It's a whole lot harder, if you look at the hurricane on the other side, to separate out, I don't know, the red number 5 and the sugar and the ice cubes and any artificial sweetener and anything else, citric acid that's in there. We have to start using much more sophisticated techniques to start teasing apart the dietary supplement that's going to become a drug. So the rest of the lecture, more or less, is going to be, we're going to walk you through Taxol. Taxol took about 40 years, so we get a whole lot to talk about. These are the two gentlemen that discovered it initially in the 70s. This is Wani and Wall, and this is actually the structure. It does not biofluoresce or anything like that, but it's a great picture. So these guys were at RTI, which is in North Carolina. It was collected in '72 from Texas bramifolia bark, which is the yew tree in the northwest United States. They didn't report the structure until '71. It took until '84 to get it to people, phase 1 clinical trials. '92, it finally gets approved for ovarian cancer. Just to give you an idea of how big a deal this is, in 2000, the sales peaked. There's lots of reasons for why. Now there's generic analogs, so but the highest it ever had made was $1.6 billion in a year for one drug. It costs between $30,000 and $40,000 for one round of treatment for a cancer patient right now. You may need multiple rounds. So what does it do? This is Dr. Horowitz. She's up at Albert Einstein School of Medicine in the Bronx. She was the woman who discovered how it actually works. And this is really what actually allowed it to finally get into clinical trials, because you can't put it in people until you understand how it works. This is a normal cell going through meiosis, doing cellular division. The microtubules come in, they grab the chromosomes have divided, and then they pull them apart into the two separate cells. Basic biology. Go back, check it if you don't remember. And you can see they all just kind of start disappearing after that's done, and cellular division goes on as normal. If you add Taxol to this, it looks like a spider web in there. All those microtubules that move around the organelles and the cells during meiosis, they stay. They get frozen in place. They can't dissolve. And that ultimately kills the cell because the cell gets filled up with them. And that's actually how Taxol does its cancer cell killing, as opposed to some of the other ones that prevent the microtubules from forming, like vinblastine and vincristine, which were the previous drugs. All right, so how would you make it? 12 kilograms of the bark was initially extracted in ethanol. Essentially, they took 12 kilograms, which is about 6 pounds, 5-6 pounds, ground it up, put it in a big container, poured alcohol on top of it, and let it sit. And then they concentrated it. This is a rotovap. This is what we use to concentrate it. Anybody who's working in our lab knows these very, very well. These are just general extracts. None of those are Taxol, the pretty picture. But Taxol was found from a screen of about, I think it was 160,000 or 16,000. I'm not sure where the decimal is in that at the moment, maybe on another slide. NCI had gone around and made tons and tons of plant extracts. It was over 10,000, and screened all of them for cancer in this cancer assay, one of which that hit was Taxol. Which brings us to the screening, 1600, sorry, when they actually had gotten to Taxol. So you have to have some assay to know what's going to be active. They went out, they collected all these plants, they made all those extracts, then they tested them in an assay called 9KB, which is a type of cytotoxicity assay. They scraped some cancer cells off of this particular type of growth. This is the inside of somebody's nose. It's got to be horribly painful. And then they grow those cells and they see if the compounds that they made in the extract kill the cells. It's done in a 96 well plate, kind of like this. And then you take your top hits and you start working with those. So the bioactivity-guided part of this is, as we simplify it or separate out the chemistry, we test after every round against some model system, which is this 9KB system. And whatever's most active, we keep working with and we ignore the rest. That's the bioactivity-guided fractionation. The reason they chose 9KB activity is because it correlates very well to a lymphoma, leukemia model. But it's a cellular assay, so it can be done a whole lot faster than having to grow up mice and inject them and then wait a while. Yeah.
How do you know you're not throwing away something valuable if an extract would, for example, not affect this specific disease?
You don't.
Yep. Would you have other studies that?
These days you'll screen in lots of different ways to try to not miss something. And when you do large libraries where they're doing thousands of screens a week, they spend several years, and I'm not kidding, developing the screening system and the models to make sure that it really is targeting what they want and they're not missing activity because of something else. Another thing you do is instead of screening just the extract, you can go ahead and separate out the extract into a couple different parts and test it in different ways that way. But ultimately, it's a little bit of a gamble. You may not find something. So they started with an extract partition. So this is a liquid-liquid partition. So they took that extract and then they dissolved it in one solvent. And they used a second solvent, shook it up. This is like oil and water in a salad dressing. They ultimately separate, and chemicals that are in each of those end up in two different places. So this is their first round of simplifying the chemistry of a natural product. They tested those two things, and it turns out that the chloroform methanol partition, not the water layer, is the one that's active. So they only work with the chloroform partition from here on out. All right, you're about to learn some old school chemistry on this one. They now have started using countercurrent chromatography again, but back in 40s and 50s, it was all the rage. This is essentially super high-tech liquid-liquid partitioning. You take your compound, you dissolve it in a solution, you put it in this weird-looking U shape, and I got a picture of the device in a minute. And you add that second solvent again so that the two solvents separate. Your compound's on the bottom, something else is floating here. The thing is on a crank, and you turn it, and the tube rotates around itself, mixing itself up, right? So you just mix two liquids that are like oil and water together. You get to the end, and if you turn it back the other way, what happens is the top layer that floats ends up on one side and the bottom layer stays on the other. And when you do that, it brings in some more of your extract. So essentially, it's like shaking up the liquid-liquid partition over and over again, but you're only doing it by cranking. What that looks like is this thing. There are lots of them. This is 60 tubes, so you put your extract down in this end, and you crank it back and forth, and it moves it down through the tubes over and over again, separating it. And you can take it off with these little stopcocks at the bottom, so you can pull off at any point some stuff from the middle or some stuff from the end. Essentially, you're fractionating it with liquids. So they used this 3 times. The first time they used 11 tubes in this system, and they found 3 of the tubes had activity in their bioactivity, or in their bioassay. Combined those tubes, put them into the second system, had 30 tubes, ran that, and they made 30 tubes. Of their tubes 8 through 18 had activity. So they combined those, put them into the last one, and this is the one that just kills me. They put it into a machine that had 400 tubes. That thing must have been like 30 feet long, I guess. I don't know. And they partitioned it across 40 different little tubes. And out of that, they ended up with 2 tubes that when they tested all 400 of them still had the activity. So they've really narrowed down what components were responsible for the activity. So for the rest of the study, that's what they're working with, are these 2 active partitions, or fractions. So you have this liquid extract that has an activity in it. How do you get the actual chemistry, the compound that's active out of it? They use a technique called tituration, which is essentially a fancy word for precipitation. You dissolve it in a solvent that it's kind of soluble in, and then you start adding a different solvent to it that it's not soluble in, and it'll crash out a solution, and then you filter out the solid, so you end up with a nice solid on your filter paper, and everything else kind of goes through. When they did this and had gone through all of this process, which was not a short thing, I'm sure, they end up with half a gram. Started with 12 kilograms, they end up with half a gram. Parth over here is giggling because he knows what's about to come next. That's a yield of 0.004%, which is tiny. Don't think that's good even in chemistry. That's really small. 12 kilograms, get a half a gram. And that is not even enough to treat one person. So at the end, when you start thinking about actually using this in patients, you have to start worrying about a supply problem. So what do you do next? That's what you got. You have a half a gram of white powder. Taxol is a white crystalline needled powder. It's active against 9 kb, which probably means it's active against leukemia, lymphoma. What can I do with that? Can I make that a drug yet?
Figure out the structure.
You got to know what it is. That's all I know. It's a white crystalline powder. The FDA is going to laugh at you and be like, yeah, so is everything else. It's an organic molecule. This is the structure elucidation part. So everything up to here has been bioactivity-guided fractionation to determine what compound is actually the active. Now we want to know what it is that actually is giving it the activity. So this is the process of determining the chemical structure of an unknown compound. there are two kind of levels of this. You have a basic 2D structure, which if you ignore the R's and S's in here, basically gives you what's connected to everything else. And then you have the relative, which is, you can add which direction things are. So rather than having two hydrogens pointing this way, you can have them pointing in opposite directions, and that gives you relative stereochemistry. But in order to really understand it, you have to have absolute stereochemistry. You have to know that if this hydrogen is pointing this way, 5 carbons away, there's an OH group that points at this angle instead of this angle. You have to know exactly what it looks like, and this gets into 3-dimensional structure determination. And that's the level that you have to get to in order to actually start calling something a drug. For those of you that had chemistry so far, they didn't have NMR. Well, they did, but it wasn't very good yet, so they had to do all this with X-ray crystallography. In order to do that, they had to take that solid and they had to make a nice crystal out of it. Who's had rock candy, like the rock candy lollipops? That's a sugar crystal. It's very square and very nice looking and regular and repeating. If you get a compound and you manipulate it just the right way and you have all sorts of luck with it, you can make it form its crystal as well. And you take that little piece of crystal and you put it on essentially a little stick, and it goes into a machine at a very specific angle, so it'll be mounted like this, and then they bombard it with X-rays. And you get this thing called an interferogram that comes out. This is actually what the data looks like. And wherever the crystal overlaps, the atoms are so regular that they literally overlap and they cast a shadow of X-rays, and that's recorded. So then you take that little crystal and you turn it a few degrees and you do it again. You take that little crystal and you turn it a few degrees and you do it again. These days a computer does all that for you, but when you get done, you take all of that data and you combine it, you do an FT analysis on it, and you can start understanding the 3-dimensional structure and what parts are there, which is great. This is like the gold standard of structure elucidation. If you can get X-ray crystallography on something, you're considered, you know what it is. Everything else you verify with X-ray crystallography. So while they didn't have these other faster techniques, the technique that they did have was an excellent tool. It's just really time consuming and really difficult. And they had this little problem. So if you read the paper, which I suggest you do, somewhere in there you'll find the phrase, a number of crystalline halogenated compounds were obtained, so they tried to make crystals of this stuff. None had properties suitable for X-ray analysis. So they tried and tried and tried, and absolutely nothing would actually work in the X-ray crystallography machine. So they had to do a bunch of chemistry on this again. And this I think is my only actual equation. So they take, this is Taxol, they did a methanalysis of it, you come up with a couple products. They take one of these products, they do some other stuff to it and add a bromine down here. This crystal they could actually do an X-ray crystallograph of. They did something else with the other products, so they actually had to break it up, do some other chemistry to it, and you end up with 2 derivatives of the original structure. So they don't know anything about the original structure, they just have 2 pieces at this point. This, if you get into the literature or a chemistry major and want to get into it, this is absolutely fascinating how they figured this out. I mean, it's just brilliant chemistry. They did have a mass spec back then, so they have 2 pieces that they kind of know what the structures are. We have to kind of know what that came from, right? We got 2 pieces, but what's the parent look like? One way we can find out information about that is we can get the mass of that compound. We can get very, very accurate mass. This is the one at Emory. This is an FTMS. We're going to pretend that it's a time of flight because it's a little quicker to explain. And the way these things work are, if Parth and Amelia are both standing over here on this wall, right? Amelia, you got a baseball. Parth, you get a basketball. You're both going to throw it until it hits that wall. Which one hits that wall first? Baseball. Why? Smaller. Yeah. And it's because things of different mass travel at different rates over the same distance like that, right? So smaller compounds are going to go through the same distance if they're given the same velocity faster than, small one's going to go faster than the big one. And if you have a very, very accurate stopwatch, is essentially what a mass spec is doing, you can time that, know the distance, and then figure out the weight of the little molecule that you just threw down the flight tube of your mass spec. To give you an idea of how accurate that is, a nickel's 5 grams, more or less. If you cut a nickel into half a million pieces, that's the accuracy you're getting. So you could take one of those 500,000 pieces of a nickel, throw it across the room, and the mass spec could tell you if it was one piece that hit the wall or two pieces. That's the level of accuracy that you need to do this next step. This was what happened when you throw Taxol into a mass spec. Now that we know what it is, there's a nice fragmentation pattern. It can show you all the different pieces that it makes. But this is the actual MS originally. Here's Taxol. It weighs 854 plus some decimal points. You can use that mass to determine an empirical formula. We won't get into this one, but you throw it into some software essentially. It looks at the isotope ratios that occur in nature versus the weight that you have. Figures out a whole bunch of possibilities and says you could have this many carbons, this many hydrogens, and this many oxygens. You come up with a nice table like this and the likelihood that your structure matches that, or your molecular mass matches that. So they had Taxol's empirical formula. They know how many carbons, they know how many hydrogens, how many nitrogens, and how many oxygens. And then they know the structure of two pieces. Can add in the degrees of unsaturation. Who's a chem major in here? 2. Do y'all both know what this is already? All right, we're going to skip over it for the rest of everybody else then. Essentially, this is going to tell you how many double bonds or rings are in your compound. If you want to learn how to calculate it, we can talk about it after class. So, at this point, you have a pure compound that cures cancer. Awesome. It has a molecular mass of 854. You have a formula of, C47H51NO14. You know there are 23 either double bonds or rings in this thing. And if you remember the structure, it's got a lot of rings. Like having that many is actually not that helpful. And you have X-ray structures for these two derivatives. So, what do you do now? See how they work individually? Together? Okay. We've already done that, right? Because we had the parent compound and it was active. We're figuring out the structure. What's the piece that we're missing? You've got two parts. You know how much the big one weighs. You got a little bit of idea of the number of double bonds. You got an idea of what the formula is, so you could look at the formulas of the two pieces and see what's missing. But if you have two pieces, what's the thing that you actually care about? Is it what the two pieces are?
What they do.
No, you already know they cure cancer. I need to know the structure of a compound. I have the structure of two daughter pieces of said compound.
Try and put them back together.
Try and put them back together. You need to know what the big piece is that they came from. As I said, they had NMR back then. For those of you that were the chemistry majors, it was continuous wave, 60 megahertz. A baby NMR these days is 300 megahertz, to give you an idea of the difference. These are all modern NMRs. This is a 600, that's a 900. But essentially what they do is they provide you information about what element is connected to what other element. So structural elements of a molecule. The two basic experiments are a proton and a carbon. We'll look at those. These 2D experiments, we have these now. They are amazingly powerful tools. They can show you all these types of connections, multiple bond connections, hydrogen to hydrogen, carbon to hydrogen over multiple bonds, through space, through bond. These are all really powerful techniques. If you're interested in any of this, there are whole classes on it here at Emory, and I'd be happy to go NMR geek out with you sometime after class. But they didn't have any of that right now, or back when they were doing this. They basically could find out information about the hydrogens and information about the carbons. Proton, it will tell you what type of proton you have, what type of hydrogen, or what the hydrogen is connected to. It's an OH group. This means it's in a ring, an aromatic structure. CH3 methyl groups, tert-butyl down there at the end. Knowing where these lines are in the chromatogram, you can use that to determine what type of hydrogen bond it is. You can also do the same thing for carbon. This is actually a depth kind of experiment, so it tells you if it's a CH2, a CH3, by if it's up or down. But you get a signal for each carbon, and where it falls on the scale, you can then determine, is it in a ring, is it a CH3 off by itself, is it, whatever you want, sp2, sp3 hybridized. This is Taxol's NMR. We are not going to go through it at all. So this is the original data. This is actually a later experiment run on it, but this is what it would look like. You can assign 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. You can assign 13 different hydrogen resonances in this. So you have the structure of 2 pieces from X-ray crystallography, and you gain information about what type of hydrogen connectivity is going on for 13 different hydrogens in this, or hydrogens on 13 different carbons out of 47. But it turns out that that information was enough to connect the 2 pieces, and this is the final structure you get. This was one of those pieces in the X-ray, if you remember, and this was the other one. So what they were looking for was this linkage right through here, and they were able to determine that from the 1D proton NMR. It took 30 years from the initial collection of the plant material until it made it into the first person in the clinic as a prescription. It took 9 years to do the structure. It was collected, and structure was in 71, so in the 60s. These days, they have come up with synthesis, so Parth, I was incorrect when I told you it hadn't been synthesized at all. In 94, there were 2 groups that did a complete synthesis of it. In 2004, 2006, some other groups have done it. It is a multi-step, very difficult synthesis to do. So it is a great academic exercise, and it can be done, but commercially, that's not how it's produced. It takes too much effort. There was a little interesting tidbit in 96. It was isolated from this endophytic fungi. Y'all done endophytes yet? Nope. So within plants, there are fungi. These are not pathogenic fungi. These are cellular fungus that live inside of plant cells or within this interstitial space of plant cells, so between the cells. And they actually are symbiotic or nothing. They may help it or they may not harm it. No, they're not quite sure right now on some of them. And they live inside the plant. Just like you have E. coli and other microflora in your intestines, plants have fungi living inside of them. Turns out in several taxus species, it's one of these fungi, this is actually the one that they first discovered, that produce Taxol. So now there's this interesting question of, does the plant produce Taxol, or does the fungus produce Taxol, and we're just isolating it when we get the bark? And that has actually become kind of a common thing now with plant secondary metabolites. Is the plant really producing it, or is the fungi producing it, or is it both of them are working on it together? It seems like both of them are working on it together a lot of times. And Emory does have a little part in this story. In 2001, Dr. James Schneider's group down in chemistry, they made a bunch of analogs and looked at different, Taxol is the trade name. And they made analogs of it and started evaluating if they worked better or not. It's actually a pretty cool paper. It does a lot of molecular modeling on it. So Emory is still involved in this sort of thing. Let's see where we go here. So as I said, it was completely synthesized in 96, but that's not really how you get it now. This is in 2011, so it's a couple years old. But essentially, it's a semi-synthesis right now. You remember the story of the U needle clippings that Dr. Quay was talking about? They went around Europe or went around Britain and asked them for the hedge clippings. So they took those needles, and you can isolate this compound from it, which is not Taxol, but it does have this basic taxane structure. And you can run it through this synthesis here, and you end up with Taxol. And that was how they commercially produced it for many years, and they may still be doing it. Bristol-Myers Squibb, BMS, 30 billion shrubs they grow to make Taxol. And Dina has a patented semi-synthesis, and Dina is a big herbal production company in Europe. They make everything from some concentrated cranberry juice extracts all the way apparently to Taxol. Then there's also the endophyte thing. There's a company that was founded that was trying to commercialize it through endophytes. I don't know how successful that was. And there's also not Taxol, but a Taxol relative called docetaxel that is more or less synthetically produced for production, and it's easier to make synthetically and almost as effective. It's actually more effective in cellular assays and tests, but not quite as effective in people. But it costs less and it's fully synthetic. So You have multiple ways that it's being produced to go into humans, but I think most of it is this semi-synthesis where they isolate a precursor and then change it a little bit. I know y'all had talked a little bit about that, but any questions on Taxol's 30-odd years? The paper y'all read is all about this. I think it was Overly's paper. Nick Overly's was one of the graduate students of Dr. Wani. We're actually postdocs. So he worked in his lab towards the end of this, or well after this, but in the same lab. So he has a pretty good perspective on it. I think I also gave y'all Wani's paper, which was written much later after it was already in production and being used, and it was kind of his historical retrospective on his discovery and Dr. Wall's work. So it's kind of interesting to see their perspective on how it came to be. Hey, Kate, would you go in the lab? And there's 2 cans of Garcinia sitting up on my bench on the shelves. So second half of this is going to be on antimalarial compounds. So we're going to totally switch gears because I want to take you into chromatography a little more than they did in the last 10 or 15 minutes here. This is a plant I worked on in grad school. It is a member of the Clusiaceae family. There's about 200 different species, 250 different species here. They're usually kind of shrubs to big trees. They're mostly tropical. Cosmopolitan means they're kind of found anywhere that it's tropical. The one that you may be most familiar with is this guy, which is mangosteen, right? Garcinia mangosteen. I actually have a couple of cans of it. I'll pass around. If I can find a can opener, we can taste it after class. I'll kick it open. It's called the Queen of Fruits, which is because the King of Fruits is often called, anybody know this one? Durian?
Oh, yeah.
Have you heard of durian? That's worth Googling sometime. Trust me. It's a good story. But it's very common to have durian and mangosteen together. And durian is a very soft, custardy kind of fruit that you eat. And you can actually dip the mangosteen, which is like an orange slice, in the custard. It's really good, by the way. The relative of that one that I worked on was actually Garcinia xanthochymus. It's about the size of a lime, but it's actually this yellow color like a lemon. It is edible. It's used in Indian cooking and chutneys quite often. It produces some really pretty little white flowers, and it stains your fingers a great shade of yellow. That's why it was used as a fabric dye, and it does taste a little bit like tamarind, but it's much more acidic. We were interested in looking at the seeds of these. Do I have it on the next slide? Yes. So, believe it or not, there wasn't a publication up until 2006 that said we looked at seeds and we looked at fruit pulp and we said the chemistry is different. Kind of makes sense, but nobody had actually done the analytical analysis to show this. So, we'd already done a lot of work on the fruit pulp. And we'd already done a lot of work on the fruit rind, so I was investigating the seeds from this plant for benzophenones, biflavonoids, xanthones, triterpenes. There's a whole lot of chemistry already out there. These are what the seeds look like. You'll notice here we did 5.6 kilograms extracted. We had a commercial partner at the time who did a big batch for us with this commercial mill that could grind it up because the seeds are really actually quite hard. And we ended up with about 418 grams of extract to do this. So again, this was a maceration where we basically were soaking it in alcohol. This is mangosteen. I'll pass one around on each side, and I'll open one after class. So after it was extracted, what was done with Taxol? They made an extract, and then what'd they do? Separated, liquid-liquid partition. Same thing here. We took a seed extract, we partitioned the water one against hexane, and then we took the leftover water one, partitioned it against chloroform, and then against ethyl acetate. We tried 3 different ways to do this, just to figure out which one would separate the chemistry out best, so that it's grouped as one group here, one group in the middle, and one group at the end. So in order to kind of see what happened next, we got to talk a little bit about chromatography. Basically a technique to separate complex mixtures, and using 2 phases, stationary and mobile phase. So something stays the same and then you pass something through it. And I'm going to pick on from the lab. Anybody want to try this one? What's the stationary phase in this gravity column? Anyone? Anyone? I got 5 of you in here. Silica. So inside of this, it's basically a fancy sand. And that's your stationary phase. You pack it in this big glass column. You see the solvent, the liquid sitting on top. You add your extract to the top, and you wash the extract through the stationary phase and collect fractions off the bottom. This is the old school way that you did it. This is the newer version. We have a machine that basically does the same thing. Your extract is loaded on top, and you can see the band going down the column right here. It's collected over in test tubes. So different compounds interact with the stationary phase in different amounts. And if they react a lot, they get stuck on the column, and they stay there a while. If they don't react much, they move with the solvent through fairly quickly. So that's how you're separating it. Things that don't react much with the stationary phase wash off quickly. Things that react highly with it react and stick, react and stick, react and stick. It take a long time to come off the column. So that's actually how you're getting the separation. The analytical version of this technique is high-performance liquid chromatography. We won't really get into this whole much, but here's the column here. Your sample gets injected on one side. It interacts with the packing in the column and then flows off the end, and there's a UV detector, so you're looking at the UV profile. We're not really collecting fractions. We're just using this to see what's in the mixture that we injected on. So if we use an HPLC, and we look at one of those partition schemes that we talked about, we can actually see what's in each of the partitions. This is hexane, ethyl acetate, and butanol. 3 partitions. You'll notice that the chemistry is pretty nicely bundled here in the middle of the chromatogram. Here it's a little earlier, and then it's kind of split at the end. We tested all 3 of these for anti-malarial activity. We'll talk just a second about that. And the hexane one was the most active, which is great because all the chemistry is kind of clustered together there. Questions on chromatography? All right, cool. This was all for antimalarial activity, not cancer, malaria. This is an assay called the Parasite lactase dehydrogenase assay. And the reason I'm actually going to tell you about this is an LDH assay is a very, very common assay used in lots of things right now, as our micro people over here are both nodding. Essentially, you are looking if a cell is alive or a cell is dead. The P, the parasite part of this, just means I'm looking at the malaria parasite, which is Plasmodium falciparum in this case. LDH is produced basically all through malaria's parasite life cycle. So at any point in the life cycle, it needs LDH because LDH actually converts pyruvate to lactate. Here's your biochemistry for the day. You have to do that in your metabolism. There's a little difference in this one, and this is what's interesting. You can have LDH that's kind of specific to an organism. So malaria lives in what type of cells? Okay. Red blood cell. So malaria, right? People get malaria, causes a fever, you can die from it. But the malaria parasite actually infects your red blood cells. So your red blood cells have LDH. They have human LDH. Malaria has LDH. Luckily, they're different enough that you can test for one without affecting the other. And that seems to be the case in a lot of systems where you can look at the LDH of one organism and kind of ignore the host cell organism, which is really helpful in a bioassay. The dark purple spots are malaria parasite. This is Plasmodium inside red blood cells. The details of the reaction really aren't that important, but essentially you grow malaria in red blood cells. It takes a couple of days, and you add your extract at some point and continue to grow it, and then you look to see versus the well that doesn't have extract, how many survived or how many died, whichever way you want to look at it. And you do that by staining it. You end up with these nice purple cell counts or nice purple cells if there was living if there was living plasmodium, there was active LDH. It stains them purple. And so you can look at how many purple cells are in there versus how many purple cells are not in there, and that'll give you a good idea of did your extract prevent the parasite from growing. That's the general gist of the bioassay. Is that clear? So we tested those liquid-liquid partitions in that bioassay we just talked about, in the LDH assay, the hexane partition basically was the only one that had activity. The azelastate and the butanol, big goose egg here. So all the chemistry that we were interested in was in the hexane partition. We took the hexane partition, and just to make sure that this was something that was really super cool that we wanted to work with, D6 and W2 clones. So we tested against 2 different types of malaria. These are the same species. They're both Plasmodium falciparum, but they're clonal lineages. So in microbiology, you can have different strains or different clones of the same organism. So cells replicate very quickly, right? Therefore, they evolve, their DNA changes, they develop characteristics at a fairly rapid rate. And you can have the same organism and just run it, and follow this, and it will develop a single trait that you're interested in. And that trait is considered a strain or a clone or a lineage off of that parent organism. In this case, this is essentially normal malaria, if there's such a thing. It responds to drugs. The W2 clone is drug-resistant malaria. So chloroquine and quinine, your kind of first-round antimalarial drugs, don't kill this. Y'all talked a lot about this, you just didn't realize it. Antibiotic resistance, right? Y'all hit that point several times in the last couple of weeks. Antibiotic resistance develops because the strain of bacteria that's infecting you is a clone or a strain that has developed resistance, and that's the one you're getting. You're not getting the one that was easy to kill. It's developed resistance, and it keeps that trait. So we had one partition that was active. It was the hexane partition. Taxol, they had the chloroform partition that was active, right? What did they do with it then? The big glass rocky apparatus, countercurrent chromatography. It's also called a Craig apparatus. It's the one with all the C's in it. That was the way they chose to then take it and break it up into small parts. We did open column chromatography, so this was that big glass column with silica in it. It's basically the cheapest, slowest, fastest, slowest, most graduate student intensive way to do it. But it works, and it works very, very well. Let's see, I made 9 partitions of this guy. Tested all of these. 2 of them still had their activity. One of them we had a lot more than the other. We'll look at both here in a second, but essentially you take one of those and we ran it through a column again, and you separate it. So are we seeing a theme here? You find activity, you separate it out into parts, you look for that activity. You find the one that has activity, you separate it out into parts, you look for the one that has activity. And you do that over and over and over again until the activity disappears, in which case you go back up a step and figure out what you lost, or there's only one thing left in there. This totally ignores a concept called synergy, which I know you'll talked about and you'll hit on again. So if that activity disappears, but there's still stuff in there, then, well, that compound wasn't the one that was responsible. If you go back up and there's a couple compounds in there and it had activity, then there's a synergistic effect going on. All those compounds were needed in order for there to be activity. The other thing that can happen is you can have 1 and 1 equals 3. So if I have one compound that's active, and let's say it's in this fraction here, and I have another compound that's active here, if I look up here where both those compounds are present, you would normally think, okay, it's going to be twice as effective. It's got 2 compounds that are active, and if you put them together, it's going to double its efficacy. What if it's 5 times more effective? And then there's something much more interesting and much more complex going on, and that's that synergistic mechanism that you talked about before. We're going to focus in the last couple minutes just on the benzophenones rather than all the chemistry that was involved in this. The paper kind of went through all of it, but we're just going to focus on half, essentially. So if we look at those active partitions, those active fractions, so if we look at these 2 right here, we actually dig into the chemistry of that using an HPLC. The first one had 4 identified benzophenones, 1, 2, 3, and 4. Here are their structures down at the bottom. It had some other stuff that, just trust me on this one, when we tested this fraction, or these fractions, those weren't the actives. It was the stuff at the end of the chromatogram. So that was one of the fractions. And the other fraction, it was much simpler. There were only these 2 compounds, fucogitin and gutiferone H. So we basically have 6 compounds that seem to have activity. We isolated those out using or open column chromatography. So again, there's that repeated separation, fractions, separation, fractions. We tested all the compounds. Of those, 3 of them turned out to actually have activity. All the other ones were just, they were nothing. Cytotoxicity there, so they had activity against malaria, but they weren't killing the host cell. That's what cytotoxicity means there. And that's really important when you read bioassay studies. Whenever you look at one of these, you want to make sure that if it has activity, somehow in the paper, in the study, they've tested to make sure that the activity they're seeing just isn't that it's killing everything, right? You want to kill your target or inhibit your disease. You don't want to kill the host. So that's an important distinction. These were all the compounds that were tested. I don't expect y'all to remember any of these structures, really any of these names. We're just doing a little bit of concept here. These were the 3 that were active. Because we had so many compounds we could look at, we were able to do a structure-activity relationship. So this is basically the relationship between the chemical structure and its biological activity. It really helps us understand what part of the molecule is responsible for the bioactivity that we're interested in. The classic example of this is you have a synthetic chemist make up lots and lots of analogs. All of these down here on the bottom, skipping back to Taxol for a second, are Taxol analogs. And you can test all of them and see, is it a little better or a little bit worse? Does this one make people, have less nausea than this one does? Does this one get into cell membranes a little bit better? So you can start adjusting the chemistry slightly. And by doing that, you can improve upon the original natural drug or make it worse. Both are possibilities. Turns out this was the basic skeleton that mattered in my work. You do not need to know the IUPAC name or the structure. It's just there. So if you kind of group them by structural similarity, here's your base structure in the middle. You see all 3 of them have the same base structure. And you'll notice, let's look at the red circles first. There's either a hydroxyl group or not a hydroxyl group there. Turns out, doesn't matter at all. There could be a hydroxyl group or not a hydroxyl group. It doesn't affect the activity. So the other part that we could look at was where the blue circle is. So this is the C8 carbon. Nothing here. This is called an IPP, isopentyl group. And it's 5 carbon chain with a double bond either here or on the end, depending. Turns out, if that's there or not there, doesn't matter either. So the other things that we could look at in this structure activity group, and I'm just trying to sort them out real quick before I have to look at them on the big screen. That one's cyclized. Okay. You can look if there's this cyclization of this chain right here. So see, it's an individual chain that connects to this hydroxyl group and makes this cycled ring. So you can look at one or the other. Turns out, you can have activity if it's a chain, or you can have activity if it's a ring structure. So that's not what's actually giving this compound its activity. Turns out if it's a chain, if you move this double bond, you don't have activity here, you do have activity here. Green square means activity. If you look at it in the ring structure, which you can't really have that double bond there, you can look at where this double bond is. And if you move that double bond around, that is where you're actually gaining and losing the activity. So these are all natural compounds. These are all compounds that actually were isolated from the plant. So we can take that information, we have the structures of these, we have a little bit of information about what structural characteristic confers activity or takes activity away as far as the malaria parasite is concerned. And you can then give that to a medicinal chemist who can then start looking at that and start doing 3-dimensional models of it and looking at target sites or even adding on different chains and manipulating the molecule a little bit and either make it better or maybe just take and say, all right, you know what, none of this actually mattered. It was just this part that was active, and we will use that part and put it onto something else, and that's how we'll have the activity. And that's kind of where the next process of the natural products drug discovery goes. You know, once you've isolated something, have an activity, and have identified what it is, you can do a little bit of this, and then it kind of moves on in the pipeline to hardcore chemical analysis and synthesis. So, to wrap up, we talked about dietary supplements and how they're different than pharmaceuticals. Remember, there are botanical drugs that are kind of the bridge between those. I think everybody gets that drug discovery takes a while and costs a lot, and we kind of walked through a whole lot of steps. So that'll give you an idea. Imagine doing that for every single compound in a drug. You have to understand it at that level. Isolation and structural determination. I'll give you a bunch of tools, kind of hit on the highlights. If you have any questions, we can do that in a second. Structure-activity relationship, again, it's used to understand what part of the chemical is most important for the activity and what part you can basically take off or manipulate without changing the activity. And then all of the stuff we've talked about today, once you're done with it, well, it's not even like 10% done. You still have to test it in mice or another cellular system. You have to get it into people and prove it's safe. And then you have to prove that, you can actually eat it and your stomach doesn't degrade it and it gets across, whatever membrane it needs to get into the target cell. There's a whole lot of pharmacokinetics and medicinal chemistry that happens after this. So this is just one piece of it. It's a very lengthy, very expensive process. All right, any questions? Any comments? All right, I'm going to go see if I can find a can opener, and we can eat some mangosteen because I don't want to take it home. And other than that, y'all are good to go.